Thank you all for joining us for the first event of the Beyond the Price Medical Lecture Series. I'm Luca Cammarata. I'm the CEO of MediCampus Europeo in Malta. The Beyond the Price Medical Lecture Series was established with the idea of bringing together Nobel Prize laureates, world-leading clinicians, researchers, and internationally respected academic voices from healthcare and academia to share their insights, experience, and innovations with our faculty, students, staff, and wider community alike. Our intention is to host several distinguished speakers each year who are helping shape modern medicine and biomedical science. This is a hybrid lecture series with some sessions held online and others hosted in person, depending on the speaker and the academic program. Although MediCampus Europeo is a young medical institution, we are deeply committed to building an ambitious international academic environment that connects our students and faculty with some of the most influential voices in modern medicines and biomedical science. For our inaugural event, I'm honored to welcome Professor Gregg Semenza, MD, PhD, a renowned scientist, researcher, and speaker. Today, he will share his insights, experience, and vision on the groundbreaking discoveries that led to his Nobel Prize in Physiology or Medicine in 2019. I kindly ask you to hold your question until the end of the talk, when we will have a Q&A session. It is now my pleasure to introduce Professor Ivan Varga, who will formally introduce our speaker. Professor Varga is visiting professor of anatomy at MediCampus Europeo and full professor of anatomy and vice dean for science, research, and PhD studies at Comenius University in Bratislava. So, Professor Varga, thank you very much for joining us and for formally introducing our distinguished speaker.
Thank you. Dear Professor Semenza, dear students, today is really a very special day, not only for you, dear students, but also for me. Like you, I have the chance for the first time to meet, learn from, and discuss with a Nobel Prize winner, and this is really exciting. For a scientist, the Nobel Prize is like the Oscar for an actor. It is the highest recognition for scientific discovery. But behind every Nobel Prize, there are many, many years, often decades of hard work, research, and dedication. Professor Semenza is an excellent example of why theoretical knowledge in biology, genetics, biochemistry, and of course anatomy, because I am an anatomist, is so important for the care of your patients. The opportunity to learn from the very best is truly important for your future. I would also like to thank MediCampus Europeo for making it possible to welcome today Gregg Semenza here. Professor Semenza received his undergraduate degree from Harvard University and earned both MD and PhD degrees from the University of Pennsylvania. He completed residency training in pediatrics at Duke University and later continued with postdoctoral training in medical genetics at Johns Hopkins. He is one of the leading biomedical scientists today. He is a professor of genetic medicine, pediatrics, and oncology at Johns Hopkins School of Medicine and director of the vascular program at the Institute for Cell Engineering at Johns Hopkins. He is also a member of the US National Academy of Medicine and National Academy of Sciences. Professor Semenza has authored more than 500 articles indexed in different databases, which have been cited over 150,000 times around the world. So it's a really extraordinary scientific output. In 2019, Professor Semenza received a Nobel Prize in Physiology or Medicine for discovering hypoxia-inducible factor 1, a key biological mechanism that explains how cells detect and also respond to changes in oxygen levels. This discovery changed modern physiology and improved understanding of cancers, cardiovascular diseases, and also human development. Dear students, you are only at the beginning of your professional journey. So you are deciding what kind of medical doctors you want to become. Please note, the best medical doctors are also scientists. Science is one big adventure. It's about discovering new things and feeling the same joy as sailors in the past. I don't know, like Christopher Columbus when he discovered new land, America. That is why I'm very happy that today's lecture will be given by a world famous physician and scientist who successfully connected clinical practice with biomedical research. Dear Professor, we are very much looking forward to your lecture, which will be followed by a short discussion. And I would already like to motivate the students to ask questions. Having the opportunity to receive answers from a Nobel Prize winner is truly a unique experience. Dear Professor Semenza, the floor is yours, and thank you very much for accepting this invitation to meet the students of MediCampus Europeo in Malta. Thank you.
Thank you, Professor Varga, for that very kind and eloquent introduction. It's a pleasure for me to speak to you all today a little bit about the history, but mostly about the present and how we're trying to move our discoveries from the bench to the bedside. So the hypoxia-inducible factors play a very critical role because they establish the balance between oxygen supply and demand. And this is a very difficult logistical problem because you have 40 trillion cells in your body, and each one of them must be supplied with adequate oxygen to meet their metabolic needs at every minute of every day of your entire life. And so the hypoxia-inducible factors regulate gene expression, and by doing so, they control the supply of oxygen because they control the production of red blood cells, which carry oxygen, and they control the functioning of the respiratory and circulatory systems that function to bring oxygen to all the cells. But in addition, at the cellular level, the hypoxia-inducible factors also regulate the consumption of oxygen by determining the relative ratio of oxidative to glycolytic metabolism within every cell. So this process is incredibly complex and requires the regulation of thousands of genes in each of your cells in a very cell type specific manner. So how did we learn about this incredible system? We started a very long time ago by asking a very simple question. We knew that the kidneys made a hormone called erythropoietin, or EPO, and that EPO was secreted into the bloodstream and then bound to erythroid progenitors in the bone marrow and stimulated their survival, their proliferation, and their differentiation into red blood cells. And of course, red blood cells carry oxygen to every cell in the body. Now, the body determines the number of red cells not by counting them, but by determining whether enough oxygen is being delivered to the tissue. And if the kidney is not getting enough oxygen, it makes more EPO. EPO makes more red blood cells, and this results in increased delivery of oxygen to the tissue. So this is a very beautiful mechanism to maintain the homeostasis or balance of the proper oxygen levels throughout the body. So if you have blood loss, EPO production will go up. If you go to high altitude where there's less oxygen in the air, your tissues will also become hypoxic, and that will increase EPO production as well. So what we wanted to understand is, how do those cells in the kidney sense the oxygen levels that are available and increase their production of EPO when oxygen levels fall? And we first identified a short DNA sequence that's located just downstream of the EPO gene. It's now known as the hypoxia response element. And this sequence has the property that we could take it out of the EPO gene, insert it into a heterologous gene, and the expression of that gene would be induced by hypoxia. We then use this DNA sequence as a way to first identify and then purify a protein that bound to it within hypoxic cells. And we call this protein hypoxia-inducible factor 1, or HIF1. So a factor just means it's some sort of protein, we think. Inducible means it's turned on in response to a stimulus, and the stimulus is low oxygen. When we purified HIF1 based on its binding to the hypoxia response element, we found that it consisted of two subunits that we called HIF1 alpha and HIF1 beta. So these two proteins come together, they bind to the HRE, and then they recruit proteins called coactivators. And these coactivators have several interesting properties. One, they interact with the transcription initiation complex containing RNA polymerase II that forms at the promoter of the gene. And this interaction serves to stabilize the formation of this complex. But in addition, these coactivators have a catalytic activity. They are histone acetyltransferases. And these proteins modify the chromatin and allow the polymerase to access the DNA and then to transcribe it into RNA. Well, after we had identified and purified and cloned the DNA sequences encoding HIF1 alpha and HIF1 beta, we were able to generate antibodies against these two proteins. And this allowed us to measure the levels of the protein in cells exposed to defined oxygen levels. And what we found is that as oxygen levels decreased from 20% to about 6% oxygen, there was a very modest increase in the levels of HIF1 alpha. But at oxygen levels below 6%, there was this dramatic increase in HIF1 alpha protein levels. So 6% oxygen corresponds to a partial pressure of oxygen of 40 millimeters, which is the PO2 in venous blood. And most of the cells in the body are exposed to between 2 and 6% oxygen. So you can appreciate then that any decrease in oxygen would occur along this steep portion of this curve, allowing a graded transcriptional response to hypoxia. The more severe the hypoxic stimulus, the greater the increase in the levels of HIF1 alpha. And because HIF1 alpha is the limiting subunit, the levels of HIF1 alpha determine the levels of active HIF1. So the obvious question became, well, how are these levels of HIF1 alpha controlled by the oxygen concentration? And this turned out to be a very interesting mechanism in which oxygen atoms are directly inserted into two different residues in the HIF1 alpha protein. These are two proline residues. And the enzymes that carry out this post-translational modification are called prolyl hydroxylase domain proteins 1, 2, and 3. And the catalytic activity of these proteins requires oxygen and alpha ketoglutarate, which is, of course, a metabolite of glucose. These enzymes use one oxygen atom to hydroxylate HIF-1 alpha, and they use the other oxygen atom to split alpha-ketoglutarate into carbon dioxide and succinate. And that makes this an irreversible reaction. Now, the insertion of this one oxygen atom into HIF-1 alpha has profound consequences because it enables the binding of a protein called the von Hippel-Lindau tumor suppressor protein, or VHL for short. And when VHL binds to the hydroxylated HIF-1 alpha, it recruits a ubiquitin protein ligase that ubiquitinates HIF-1 alpha, and that's a signal for the protein to be degraded by the proteasome. So under normoxic conditions, when oxygen is available, this reaction is carried out and HIF-1 alpha is degraded. Now, under low oxygen conditions, this reaction is inhibited. And HIF-1 alpha protein that's not hydroxylated cannot be bound by HIF-1 alpha, by VHL, and it rapidly accumulates in hypoxic cells. Now, in addition to the hydroxylation of these two proline residues by the PhDs, we had identified a protein that we called FIH1 for factor inhibiting HIF-1 because we had found it interacts with HIF-1 alpha and interferes with its transcriptional activity. And it turned out that FIH1 is also a hydroxylase. It hydroxylates an asparagine residue in the transactivation domain. And this hydroxylation, which involves the same sort of biochemistry as the prolyl hydroxylases, this hydroxylation prevents the binding of the coactivators that I told you about earlier. Again, under hypoxic conditions, the hydroxylation reaction is inhibited, and now the coactivators can bind, and HIF-1 alpha is competent to activate transcription. So you can appreciate that both the half-life of the protein and its activity as a transcription factor are being modulated by these oxygen-dependent changes to the protein. And so this provides a mechanism to directly transduce changes in oxygen availability to the nucleus as changes in HIF transcriptional activity. And this has profound consequences because, as I mentioned, we now know not just of one gene that's regulated by HIF-1, but thousands of genes. And these genes have many functions. Some of them, like EPO, serve to increase oxygen delivery, whereas others help cells to survive oxygen deprivation. And as I mentioned, one of the critical adaptations to hypoxia is the switch from oxidative to glycolytic metabolism. And HIF-1 controls the expression of glucose transporters and glycolytic enzymes that allows it to determine the relative ratio of oxidative to glycolytic metabolism in every cell in your body. Well, we started out with HIF-1, but after we had identified the DNA sequence of HIF-1 alpha, it was clear that there was a related gene called, that's not encoding a protein that's now called HIF-2 alpha. So this is encoded by a different gene, but it's structurally and functionally very similar to HIF-1 alpha. So it's oxygen regulated, it dimerizes with HIF-1 beta, it binds to HREs. But some genes are controlled by HIF-1, some are controlled by HIF-2, and some are controlled by both. And we don't quite understand the basis for that selectivity yet. So still plenty to understand with regards to the regulation of gene expression in response to hypoxia. Well, unfortunately, this beautiful homeostatic system is hijacked by cancer cells. And this is where our current work comes in, because for the last 15 years or so, we've been studying the role of HIFs in cancer progression and understanding how we might intervene therapeutically to more effectively treat cancer. And this is going to be the main topic for my presentation today. Well, I mentioned to you that HIF1 and HIF2 have both unique and shared gene targets in every cancer cell. And unfortunately, in every cancer cell, the particular genes on these lists is different. So one of the amazing things about this system is its plasticity. And what I mean by that is that the response to hypoxia is unique in every cell, both every normal cell in your body and also every cancer cell. You can think of the HIFs as simply sending a message to the nucleus, there's hypoxia, respond. But how the cell responds is dependent upon how it's programmed, which is dependent on all of the other transcription factors in the cell, which determine which genes are on. Because in general, the HIFs don't turn genes on and off, but rather they take genes that are on and increase their expression. And this is a study that we did some time ago where we knocked out either HIF1 alpha or HIF2 alpha in a breast cancer cell line, or both, or neither. And then we injected these breast cancer cells into a mouse and measured the growth of the tumors that formed over time. And you can see that when both HIF1 alpha and HIF2 alpha are expressed, the tumors have the greatest growth rate. When either HIF2 or HIF1 alpha expression is silenced, the growth rate is impaired. And when both HIF1 alpha and HIF2 alpha expression are knocked down, we have the greatest impairment in tumor growth. And this has been shown in many different cancer types. So we conclude from this that both HIF1 and HIF2 are contributing to cancer progression, and presumably that's because of the fact that they have these unique and shared gene targets. Well, this is a picture of a particularly aggressive cancer. It's a very aggressive cancer because the cells are dividing so rapidly that there are cells that are too far away from a blood vessel to receive enough oxygen to survive. And so this white area is where cancer cells have died because there was no oxygen available, because all of these cells had consumed that oxygen. And you might say, well, what's the matter with that? We like dead cancer cells, but the problem is these brown cells here. And these are not actually cells that are brown, these are nuclei. And the nuclei are brown because we've stained with an antibody that we developed against HIF1 alpha. And you can see that the cells that have very high HIF1 alpha are surrounding the areas of necrosis. What does that mean? That means these are the cells furthest away from a blood vessel that have the lowest oxygen concentration but can survive. And what we know is that these cells can not only survive low oxygen, they have increased ability to survive chemotherapy, radiation therapy. These cells have increased ability to invade and metastasize through the body. And most of the cancer drugs that are used in patients target rapidly dividing cells that are present near blood vessels where the oxygen is available. And there haven't really been any drugs designed to kill these hypoxic cancer cells. And we set out to develop a cancer therapy that targets these cells that have very high levels of HIF-1 alpha. And the use of the antibodies to perform immunohistochemistry like this in many different cancer types by our lab and many other labs has shown that in almost all of these cancers, patients who have very high levels of HIF-1 alpha in their primary tumor biopsy are much more likely to die of cancer than patients who have low levels of HIF-1 alpha in the biopsy. And as you can see, this applies to every common cancer type, and there are now over 100 published studies that say increased HIF-1 alpha increases the risk of cancer mortality, highlighting the important role of HIF-1 in cancer progression. Well, why is that? It's because the HIFs control the expression of genes that contribute to every critical aspect of cancer progression. And I'm going to focus on just one of them in this talk, which is the ability of cancer cells to evade killing by the immune system. So cancer cells are very abnormal. They have many mutations that result in abnormal proteins. These abnormal proteins help them to grow, but they can also be detected by the immune system, and that would trigger the immune system to kill these abnormal cells. However, over time, cancers develop mechanisms that allow them to avoid being killed by the immune system. And one of the major research goals has been to understand that process and then to figure out ways to reactivate the immune system to kill cancer cells. So the particular types of cancer cells, of immune cells that function to recognize cancer cells and kill them, there are several types, and one of these are T cells that have a protein on their surface called CD8, and so they're called CD8 positive T cells. And when these cells recognize a cancer cell, they can kill that cell. Unfortunately, the cancers make a protein called PD-L1. And PD-L1 binds to a protein on the T cell called PD-1. And when this interaction occurs, it triggers the T cell to become what's called exhausted, meaning it's not capable of doing anything in response to the cancer cell, or even killing the T cell. And this is a major mechanism by which the cancer cells can evade killing by the immune system. So when this system was identified, it became obvious that if this interaction was interrupted, it would prevent the cancer cell from inactivating the T cell. And so a strategy for treatment called immune checkpoint blockade was developed, which was the development of antibodies that bind either to PD-1 or to PD-L1 and disrupt this interaction. There's a third type of immune checkpoint blockade that targets another protein on T cells called CTLA-4. And when these antibodies are successful in restoring the ability of the immune system to kill cancer cells, the response is very dramatic. Patients with extensive disease throughout their bodies have essentially been cured of their cancers because the immune system can find cancer cells wherever they are and destroy them. Unfortunately, this is the minority of patients who are treated with these drugs. Less than 20% of patients show that kind of a complete response. Why is that? Why do so few patients respond in this manner? And the answer is that the cancer cells have many other mechanisms by which they can evade killing by the immune system. And I'll just mention one here, which is a protein called CD73. This is a protein on the surface of the cancer cell. It converts a small molecule called AMP to a small molecule called adenosine. And this adenosine can bind to a specific receptor on the T cell and trigger exactly the same response that's triggered by the interaction between PD-L1 and PD-1. So you can appreciate that even if this interaction is blocked, if the cancer cell expresses CD73, it still has a mechanism to inactivate the T cells. And you can appreciate that cancer cells that express both of these proteins would be resistant to a treatment that targeted either one of them. And, we can just take breast cancer cells, expose them to oxygen for 3 days, to low oxygen levels, for 3 days. And we can identify cells that express both CD73 and PD-L1 on our cell surface. So it's clear that we need more general strategies for reactivating the immune system. Another way that the immune system can kill cancer cells is to eat them. So there are immune cells called macrophages that can perform what's called phagocytosis, where they literally envelop the cancer cell and eat it. Now, the cancer cell has a strategy for dealing with macrophages as well. It expresses a protein called CRT, which interacts with a protein on the macrophage called LRP. Okay, this triggers phagocytosis, but the cancer cell can express a protein called CD47, which interacts with the macrophage and blocks its ability to perform phagocytosis. So just like these two proteins block the ability of T cells to kill cancer cells, this protein blocks the ability of macrophages to kill cancer cells. However, you'll notice that all three of these proteins, their expression is increased in response to hypoxia through the activity of HIF1 and HIF2. So if we block HIF activity, we block the increased expression of all three of these proteins. Well, this suggested to us and many other people that developing drugs that inhibit the HIFs might be a useful therapeutic strategy. Now, the first drug that binds to one of the HIFs is a drug called belzutifan. And you can see it's a small molecule shown here. And it can bind directly to, you can see this little crevice in the HIF2 alpha subunit. And when the drug binds here, it alters the structure, the shape of the protein, so it can no longer interact with HIF1 beta. And if these two proteins cannot interact, they do not form a functional transcription factor. So this drug, which can only bind to HIF2 alpha, cannot bind to HIF1 alpha because this crevice is not exactly the same in HIF1 alpha. It's effective in treatment of kidney cancer, a kidney cancer called renal cell carcinoma. And this is one of the early studies that was done in patients who have renal cell carcinoma who had basically failed existing therapies. And what each one of these bars is a different patient's cancer, and what's being plotted is the change in the size of the tumor from when the treatment started. So anything above the line means the tumor size increased. Anything below the line means it decreased. And these dotted lines are the lines basically where the increase or the decrease is actually clinically relevant. So you can see that there weren't many tumors that increased in size once the patients were started on belzutifan, but there were many patients whose tumors decreased in size. And then many patients whose tumors basically stayed the same. They really didn't change very much. And so this is what you want if you have cancer. You want your tumor either not to grow or to get smaller. And in this study, that was 80% of the patients, which was very remarkable for a trial where the patients had already failed all of the existing therapies. So this drug was approved by the U.S. Food and Drug Administration for the treatment of renal cell carcinoma. And it's been a very valuable addition to the therapeutic regimen for treating this cancer. But I told you that for many cancers, both HIF1 and HIF2 play a critical role. And so it's been very interesting that there have been no subsequent approvals of belzutifan for other more common solid cancers. And we think this is because our renal cell carcinoma is an outlier in that the tumor progression in many of these kidney cancers is driven exclusively by HIF2. In fact, some of these cancers even lose the HIF1 alpha gene. Whereas in other cancers, as I've tried to emphasize, HIF1 alpha plays a major role, and inhibition of HIF2 alone is not sufficient for a maximal therapeutic response. So it was our contention that it was very important to develop a drug that inhibited both HIF1 and HIF2 to effectively treat other cancers beyond renal cell carcinoma. And this just emphasizes the fact that all of these more common cancers, these are the number of patients who die of these cancers per year in the United States alone. And you can see that there are 14,000 deaths due to kidney cancer and over 350,000 deaths due to these 7 cancers above kidney cancer in terms of the mortality. And there's fairly good evidence that in each one of these, both HIF1 and HIF2 are contributing to cancer progression. So what strategy did we take to identify a drug that inhibits both HIF1 and HIF2? Well, we started with the fact that the crystal structure, so you remember I showed you a structure of HIF2 alpha and HIF1 beta. So we started with that structure and asked, are there other spaces in the protein where a small molecule could sit, like that space I showed you that's occupied by belzutifan? And so we teamed with a group who developed a computer algorithm that is very good at identifying places where molecules could bind and then predicting specific molecules that might be able to stably interact with the protein. And then so the algorithm gave us a list of hundreds of compounds, and then we purchased those compounds and tested them one by one to see if they inhibited the ability of HIF1 or HIF2 to activate transcription in cancer cells. And we identified 2 compounds that were able to inhibit both HIF1 and HIF2. So we said, that's great. but can we make these compounds better? Can we make them able to inhibit the HIFs at lower concentrations? And can we give them better drug-like properties? Like rather than injecting them, can we make a compound that can be given by mouth? And so we then made these derivatives and then tested whether they could affect tumor growth in mice. And we also established the mechanism of action. And we settled on a small molecule that we call S9N. And S9N can bind to a region in HIF1 alpha and HIF2 alpha that are very similar. So the amino acid sequence in this part of the proteins are over 90% similar. And this explains why S9N can bind to both. The binding of S9N prevents the interaction of the alpha subunits with the beta subunit. So like belzutifan, you can't form a functional dimer. But S9N has an additional property, which is it triggers the destruction of HIF1 alpha and HIF2 alpha. So the proteins are completely eliminated from the cancer cells. And as I mentioned, we tested the ability of S9N to inhibit tumor growth. So this is an example of a brain tumor called glioblastoma multiforme, or GBM for short. GBM is a terrible disease. Here you can see a tumor growing in the brain of a patient. This patient would require brain surgery to remove as much of the tumor as possible. It's not possible to remove all of it. The patient would then receive radiation to the brain and then would be treated with a chemotherapy drug that attacks dividing cells, but not just dividing cells in the cancer, unfortunately, but dividing cells in your hair, dividing cells in your GI tract, resulting in nausea, vomiting, loss of hair, all of the things that you associate with chemotherapy. And in the end, these patients only live about 15 months after diagnosis, despite those terribly demanding treatments. So here we've injected these GBM cancer cells into a mouse. We've allowed the tumor to grow, and then we start treatment either with S9N or the solution that we use to deliver the drug. And you can see that as soon as we start treatment with S9N, the tumor growth stops. And we see this in pretty much every model of cancer that we test in mice. When we start the treatment, the growth of the tumor is inhibited. Well, in those models, we inject a large number of cancer cells all at once. There's another type of model where the tumors form gradually in the animals over time because we engineer the mice to express a tumor-causing protein in a particular cell type. So in this model, we're expressing this oncoprotein in the cells of the breast. And as a result, the female mice all develop tumors over time. And by 7 weeks or so, you can palpate the tumor in the mammary gland of the mice, and these tumors then gradually increase in size. So we decided we would start the treatment just before the tumors become palpable. So in other words, the tumors have been forming during the entire life of the animal, and we're going to intervene kind of at the last moment. And we treated the animals with either the vehicle, just the control solution, with belzutifan, the HIF2 selective inhibitor, or with S9N, the dual HIF1, HIF2 inhibitor. And we looked at the survival of the animals over time. And you can see that the vehicle-treated animals have all died by day 94 of life. And on that day, none of the animals treated with S9N even have a palpable tumor. And you can see that these animals do eventually die of the tumors, but they survive much longer than the control mice or mice treated with the HIF2 selective inhibitor, belzutifan, which, as you can see, does not have a significant effect on the survival of the mice. This is a colon cancer model. This is, again, one of the transplantable models where we inject a large number of cancer cells, allow a tumor to form, and then begin treatment either with the vehicle, a HIF2 selective inhibitor, or S9N. And again, you can see that the HIF2 selective inhibitor has a partial effect on tumor growth, and the S9N inhibitor has a much greater effect in terms of inhibiting tumor growth over time. So at the end of the experiment, we took the tumors out of the animals and then stained sections of the tumors for blood vessels. Because we know that one of the ways that the HIFs control oxygen delivery is by stimulating blood vessel growth. And you can see that in the tumors from vehicle-treated mice, the blood vessels, which are stained brown, you can see there are very large blood vessels in these tumors. Whereas in the mice treated with S9N, the tumor blood vessels are very small. In most of them, you cannot even see the lumen, the opening in the middle of the blood vessel, because they're very small. So as a result, of course, the delivery of oxygen and nutrients to the tumor is greatly reduced. And we think that this is an important mechanism by which S9N inhibits tumor growth. And then you can see for the HIF2 selective inhibitor, the blood vessels are smaller than in the vehicle, but significantly larger than in the S9N treated tumors. And here we've quantified the total area of blood vessels in sections from mice treated with vehicle with the dual HIF inhibitor or with the HIF2 selective inhibitor. And you can see that treatment with the dual HIF inhibitor results in the greatest decrease in blood vessel growth. Well, I mentioned that, we're interested in the ways in which the HIFs allow cancer cells to evade killing by the immune system. And so we wanted to know whether treatment with S9N would improve the response to immune checkpoint blockade, treatment either with an anti-PD-1 or anti-CTLA-4 antibody. So we've chose 5 cancer models which were known to be resistant to immune checkpoint blockade. And we allowed the tumors to form and then treated with immune checkpoint blockade alone or in combination with S9N. And you can see that immune checkpoint blockade alone resulted in a low response. Actually, this is specifically a complete response. What do we mean by a complete response? We mean that the tumor is completely eradicated. The animal had a tumor that we could palpate, and now it's gone. It means that after we stopped all treatment, the tumor did not come back. And even if we injected more cancer cells into the animal, no tumor formed because the immune system was able to recognize the cancer cells and kill them. And you can see that in every one of these cancers, when we added S9N in addition to the immunotherapy, we saw a dramatic increase in the number of animals with a complete response. And if we could achieve this kind of an increase in the response in the clinic, it would mean hundreds of thousands of lives saved potentially. Okay, well, I told you the HIFs are very important in the body. Is giving us a HIF inhibitor safe? So to answer that question, we treated animals with S9N, we treated them twice a day, whereas the tumor models we treated once a day. So we're giving twice the therapeutic dose for 40 days. And we noted that there was no change in the appearance, behavior, or body weight of the animals, no change in blood chemistries indicating normal liver and kidney function, no change in platelets or white blood cell counts indicating normal bone marrow function, and no change in histology across 8 organs. So it appears that in mice, S9N is safe. And we increased the amount of drug that we gave the animals to see, is there some dose at which it no longer becomes tolerable? And basically, we were limited by the amount of drug that we could give the animals by mouth. We could not really establish a dose that caused a toxic outcome in the mice. So the drug seems to be quite safe. How do we want to use this drug? Well, we're interested in treating cancers for which there's no effective therapy currently. And this is a list of some of the cancer types that are resistant to therapy, including immunotherapy, and where we think adding S9N in combination with immunotherapy may allow for a response and an increase in survival of these patients who have very short survival times, each of these cancer types. So just to summarize what I've told you, S9N is this first-in-class dual HIF1, HIF2 inhibitor that binds directly to the HIF1 alpha and HIF2 alpha subunits and causes them to be degraded. It inhibits tumor growth, vascularization, invasion, and it sensitizes a broad range of cancers to respond to immunotherapy. We can treat the animals once a day by mouth, and the drug is effective. As I've shown you in 2 different models of breast cancer and colorectal cancer, it appears to be more effective than an inhibitor of HIF2 alone. And as a result, we think it addresses a major unmet clinical need and that it will be poised for translation to a much larger patient population than belzutifan. So we hope that for many patients that this drug could be a lifesaver. Sadly, there are 10 million cancer deaths every year worldwide. And of course, these patients die because there's no effective therapy for their cancer. And we believe that targeting the HIFs will be a major advance in treating cancer. So I just wanted to show you the people in the lab who did the work. First of all, we collaborated with Alex McCarroll's laboratory at the University of Maryland, and his lab has over a decade developed this computer-aided drug design strategy that started us on our way. Tina Huang is a really talented tech in the lab. She's originally from Taiwan, and she screened over 40 different human and mouse cancer cell lines to screen them for effective inhibition of HIF activity by the compounds that we had developed. Yusang is from Korea, and he's the very talented chemist who synthesized S9N as well as hundreds of other chemical compounds during our drug development process. And Shaima is a very talented research associate from Canada, and she really was the driver of the program and performed all of the animal experiments. So I thank you very much for your attention, and I'd be very glad to try to answer any questions.
Dear Professor, thank you very much for your lecture. congratulations to your results. And I just would like to explain students that this type of research is an excellent example of so-called from bench to bedside. So how professor translated his research from laboratories through animal experiments to improvement of diagnostic thanks to immunohistochemistry and also drug development. So it was really a very, very nice example of translational research, though, for everybody. Before I will open the discussion, maybe I will have one general question for you. What can you recommend to our students in his and her careers at the very, very beginning stage? So how can a normal, ordinary student of medicine become a successful scientist? So what is more important according to you, the hard work or good luck, maybe good teachers or knowledge or everything together?
Yes, everything together. I was very fortunate because I was inspired by my high school biology teacher, and I developed a passion for science based on the way that she told us about not science, but about scientific discovery and the excitement of that process. And I was very fortunate to have great mentors all along the way. And I'd like to mention that I failed many times along the way and had many moments of despair and doubt. But one of my mentors would always say, search and research. And persistence is a necessary quality for successful scientific research. When things don't go right, we view them as challenges, obstacles to be overcome, not great tragedies. I think it takes all of these things, and certainly in my case, good luck was definitely one of them, to have a career, a successful, what would be described as a successful career in science. But I could just say, that for me, it's such a satisfying career because I have tremendous freedom to follow my interest and answer questions using whatever techniques, I want to use. And as long as I'm successful, I'm pretty much left alone. And so that kind of freedom and creativity and enjoyment of the work, I think we're very fortunate as scientists because many people don't have that kind of a satisfaction from their career.
Thank you. Thank you for your recommendations. And now the discussion is open. So students, if you have some questions, please feel free to ask Professor Semenza.
Hello. Hi, Professor and Professor Semenza. My name is Anas Kanji, and this is my colleague Paula Posiute. And we have some questions that we would love to ask you. But first of all, we would like to thank you very much for being here today, for your very good presentation, which was very easy to understand even for us, and for your inspiration. Thank you very much.
Thanks for your kind feedback.
Thank you. So my first question that I'm itching to ask is related to the Warburg effect. So I'm thinking in the treatment here. You've mentioned that we are targeting treatments that target both HIF1 and 2, like S9N, and you also mentioned belzutifan, which only targets HIF2. But if the cancer has an alternative pathway, which would be the Warburg effect, do we need to also focus our treatments in inhibiting this effect, which would be aerobic glycolysis, which would grant the energy to the cancer cells?
Right. So as you may know, if you've ever seen a chart of metabolic pathways, they're very complicated and nonlinear. So it's more like a web of different reactions. And as a result, you can block one of these pathways and there are alternative mechanisms to get around the block, right? So this makes targeting metabolism very challenging. Now the HIFs, because they're affecting the expression of many genes in a pathway. So, for example, coordinately regulating all of the glycolytic enzymes and glucose transporters, they can have profound effects on metabolism. And because we're giving the drugs systemically, we can hopefully get to all the cancer cells that way. So metabolism is undoubtedly one way in which HIF inhibitors exert a pressure on the cancer cell. I mentioned another, which was angiogenesis, right? To deliver oxygen and glucose, right, to the cancer cells, because regardless of their metabolism, they need glucose to burn. And then effects on the immune system to protect the cancer cells from killing by immune system. So this is the power of the HIFs, is that they're operating, at each of these critical aspects of cancer progression. The HIF inhibitors are having a role. And we believe it's that unique ability to target so many pathways that are important to the cancer cell that we can have this profound effect. Still, though, probably not even by themselves, though, right? Because as you saw, we could block tumor growth when we just used a HIF inhibitor, but we couldn't eliminate the tumor, right? It was the combination of immunotherapy and HIF inhibitors that allowed us to eliminate the tumor. And I believe that this will be the most powerful use of these compounds, is that they will be added to existing therapeutic regimens and make those regimens work better. And that includes immunotherapy, but I think it includes many of the targeted therapies as well. Because it turns out that many of the signaling pathways that are turned on in cancer cells, one of the effects is to increase HIF activity in an oxygen-independent manner. And so when you inhibit those pathways, you actually have an effect on HIF activity, and you cause hypoxia within the tumor, which just increases HIF activity again, right? And so you need to block. And the classic example is a VEGF inhibitor, right? Why don't they work for cancer? They reduce primary tumor growth because they block vascularization, but they make the tumor much more hypoxic and therefore much more likely to invade and metastasize. Not a good thing for a cancer therapy to do, right? And this is why they have not been successful as cancer therapeutics. If you combine them with a HIF inhibitor, that might be a different story. So we really think that these dual HIF inhibitors will have many roles as a way of improving existing therapies. And so far we've done experiments in mice. That's just one step along the way. We know that what the challenges in patients are much greater. But unfortunately, the only way we can find out is to perform clinical trials. So we're trying to get to that stage of being able to perform a clinical trial. We first have to do additional safety studies in larger animals to be sure, or to have a greater assurance, let's say, that the compounds will not have a severe adverse effect in patients. But unfortunately, even that we only know by performing the first early studies, where we slowly increase the amount of drug that's given to patients, so that we're sure that we won't give a large dose that might have a very severe adverse effect. So that's sort of where we are. And could the HIF inhibitors in combination with a drug that targets metabolism, could that be a good combination in certain tumors? Certainly, it may be. It may be. So I will leave that up to you to find that
hopefully one day
that metabolism targeting drug that will work well in a combination with a HIF inhibitor.
Thank you.
Because obviously, yeah, we publish our data with the hopes that other people will try to extend our results in areas where, of greatest interest to them.
Absolutely. Thank you very much. My second question, if you allow me, Professor, I would like to mention that Alzheimer's disease has been increasingly linked with hypoxia signaling. So I was thinking, if is it possible to use the signaling as a protective more than destructive power in neurodegenerative diseases?
Yeah, that's a very interesting question. This is really the next frontier, right? Is neurodegenerative disorders, because we really haven't been able to make a dent there at all therapeutically. And of course, it's the most complicated organ in the body. So yeah, it seems as though, there is a decrease in perfusion associated with Alzheimer's brain changes. But unfortunately, when you start to look at the details with regards to the HIFs, you can kind of make a story in either direction. And I think this, again, reflects the vast number of target genes in different cell types. And we have to figure out what would be the net effect, right, of dialing up or dialing down the system. And I'll give you an example. There were studies showing that the HIFs are important for the function of the T cells. So we said, oh my God, we give a HIF inhibitor, we may actually have it, a bad effect on the immune system. So what would be the net effect? Well, we know that one of the things the HIFs do is prevent the T cells from even going into the tumor. So clearly, if they can't go into the tumor, it doesn't matter, whether they're alive or dead, right? The first thing is you got to get them in there. And it appears that the net effect of HIF inhibitors, when given to an animal with cancer, is to promote the ability of the immune system to become reactivated. And I think that probably the complexity of neurodegenerative disorders, it may require a similar empirical test, right? What is the net effect of this complicated, network of genes and different cell types, right, that are being affected by these disorders? So yeah, I view as this, as the area as the number one challenge for the next generation. Is to tackle neurodegenerative disease.
We'll do our best. Thank you very much. I'll leave the floor to my colleague Paula. She has a couple of questions as well.
Good evening, everyone. Thank you so much for this opportunity. Thank you for the lecture, Professor. It was very, very interesting. And I would like to continue on the same note as my colleague has started. So I will continue with cognition and neurodegenerative diseases. So given that the brain is very sensitive to oxygen deprivation, what do you think is the most underexplored connection with HIF signaling and neurodegenerative diseases and cognitive function?
Yeah, so I think one of the interesting areas is the regulation of stem and progenitor cells. So there's some, limited ability of cells in the brain to regenerate, and cells that are located in specific locations or niches in the brain. And there's evidence in general that stem cells seem to occupy hypoxic niches in the body. And we know one of the things the HIFs do in cancer, unfortunately, is they promote the specification of cancer stem cells. And we think that's probably why there are hypoxic regions in tumors, is because that's where all the stem cells are being born. So I think that this area of stem progenitor cells and the role of the HIFs in those cells in general is an interesting one. The other thing that we've noticed when we were studying the cardiovascular, the vascular response to ischemia, was that the vascular response is impaired by aging. And of course, Alzheimer's is an aging-dependent disorder, right? So the question is there an age-dependent impairment in HIF activity that's contributing to the pathogenesis of those diseases? So, yeah, you've asked me questions in an area that I definitely know very little about. I'm sorry that I'm not able to give a more astute answer, but those are some of the areas that I kind of view as interesting for future research.
Thank you so much, Professor. That's a very interesting answer that you gave. And my second question doesn't have to do much with cancer or your research, but it's interest of mine. So, because studies suggest that prenatal hypoxia is a risk factor for schizophrenia, and there's growing evidence linking hypoxia with other psychiatric disorders like depression. Do you think targeting oxygen sensing mechanisms could open new ways and possibilities for psychiatric treatment?
Wow, so you did find something I know even less about than neurodegenerative diseases, which is psychiatric illness, and prenatal influence. So you're really testing me now. That's a very interesting question. We know, so neurodegenerative disorders, psychiatric disorders, we know these are very common. Why are they very common? They're very common probably because there are many etiologies, right? Many ways you can get to this final endpoint, which we call depression or schizophrenia, whatever we decide to call it. Just like there are many types of cancer. And the challenge is for each one of these, obviously the driving force may be slightly different, and we have to consider evidence from different sources, like the evidence suggesting that prenatal hypoxia, may have a adverse effect, and we certainly know that. The fetus, of course, is already in a hypoxic environment, right? So the PO2 that the fetus sees is only about 40 millimeters of mercury compared to 100 in arterial blood in afterbirth. So it's very interesting, right, that the fetus develops in a very hypoxic environment relative to the postnatal environment. And so you have to wonder, that must be beneficial in some way, right? For everything, right? So our life and the function of our cells is exquisitely focused on a certain range of physiological PO2, and oxygen levels above that or below that result in pathology, right? So the delivery of oxygen is very precise, because we know that oxygen can have damaging effects, right, through the formation of free radicals and oxidative damage. So we know that the body maintains those oxygen levels very precisely. At all times, right? Before and after birth. And it's a real challenge to think about, well, why are they so low before birth compared to after birth? And then what happens during the development of organs, during the life of the organism. These are all things that we need to study, or you will need to study in greater detail.
Okay,
Thank you so much.
Thank you.
For your
The
questions.
time is over, but according to also these questions, we can see that in the future we will need one more lecture from you, probably about hypoxia in prenatal development, embryonic development. It will be really very interesting and really up to date. So, dear professor, on behalf of our students and teachers of MediCampus Europeo, I would like to say thank you very much for your time, for your lecture, for this very interesting discussion, and we hope that we meet together in the near future.
Thank you, Professor Varga. It's been a pleasure.
Thank you very much. Have a nice day. Goodbye.
Bye.